Imagine if you could have a biological machine that could just print out copies of any gene you wanted. Well, back in the 1970s, that's exactly what scientists built. It was this tiny circle of DNA, a little biological program called a plasmid, and it became an absolute legend. So today, let's talk about the story of pBR322, one of the original workhorses of genetic engineering. A dream machine. I mean, that's a pretty bold claim for a microscopic loop of DNA, isn't it? But that is exactly how scientists in the late 70s saw this thing. This wasn't just another small discovery. It was a total revolution packed into a tiny biological tool called a plasmid. So what on earth made it so special? To really get why pBR322 was such a game changer, we've got to go back in time to the 1970s. You had these molecular biologists who were right on the edge of a massive breakthrough. They had just figured out this incredible new power, but it came with an equally massive challenge. So, scientists had just discovered these amazing things called restriction enzymes. The easiest way to think of them is as molecular scissors. They're these proteins that bacteria make that can scan a super long strand of DNA and cut it. But here's the kicker. Not just anywhere. They cut at very specific, predictable spots. And this right here is what that precision looks like. You have an enzyme, say EcoRI, and it will just chug along the DNA until it finds its exact target sequence, in this case, GAATC. And boom, it makes a perfect, clean cut. For the first time ever, scientists could reliably snip out a specific gene. But this incredible new ability immediately created a brand new problem. Right, so you've got one copy of a gene. That's great, but it's not very useful. If you want to study it or, get bacteria to start producing a protein like human insulin for you, you don't need one copy. You need millions, even billions of copies. What they desperately needed was a biological photocopier, some kind of vehicle to carry that piece of DNA into a living cell and then trick that cell into making tons of copies for them. And the answer to that problem, that photocopier they needed so badly, it finally arrived in 1977, and it came out of the lab of Herbert Boyer over at UCSF, which at the time was basically the epicenter for this radical new field of recombinant DNA technology. This was it. The solution was a small, circular piece of DNA called a plasmid. But, and this is the important part, it wasn't some plasmid they just found lying around in nature. Oh no, this one was engineered. It was built from the ground up, specifically to be the perfect vehicle for cloning genes inside the bacteria E. coli. And the name itself actually tells the whole story. The P is simple, it just stands for plasmid. BR are the initials of the two postdocs who actually built it, Francisco Bolivar and Raymond Rodriguez. And 322? Well, that was just the lab's number for it, to keep it separate from all the other plasmids they were tinkering with. Super simple, super practical, and it was about to become one of the most famous names in all of molecular biology. So, what was actually under the hood of this thing? Let's take a closer look at what made this dream machine tick. What were the key design features that made pBR322 so incredibly good at its job? Okay, so this is the blueprint. Now, it might look a little complicated, but this map of 4,361 base pairs shows everything a scientist back then could have wanted. It was elegantly simple, really, and it had three absolutely essential parts that made it the perfect tool for cloning. All right, first you have the origin, which stands for origin of replication. This is basically the engine. It's the sequence that tells the E. coli cell's own machinery, hey, you, copy me, and copy me a lot. Then you have these two things called selectable markers. You can think of them like secret passwords for survival. The AmpR gene gives the bacteria resistance to the antibiotic ampicillin, and the tetraR gene protects it from tetracycline. And this little feature is absolutely crucial, as you're about to see. And finally, the third brilliant thing was its incredible versatility. Sprinkled all through its DNA sequence were over 40 unique docking sites for all those different restriction enzymes, the molecular scissors. Just think of them as a whole bunch of universal USB ports, giving scientists amazing flexibility to cut the plasmid open and plug in whatever gene they were interested in. So you have this perfect little tool, but how do you actually use it? You've got the plasmid, you've got your gene. How do you put it all together to start making billions of copies? Well, the process is a classic lab experiment called bacterial transformation. The process itself is almost like a simple recipe. First, you take your E. coli cells and you treat them with some chemicals to make them competent, which just means they're ready to soak up some foreign DNA. Then, you mix these cells with your pBR322 plasmids.
After that, you give them a quick heat shock, a sudden change in temperature that punches these tiny holes in their membranes, just long enough for a few plasmids to sneak inside. You let them recover for a bit, and then you spread them all out on a petri dish to grow.
But here's the catch. This whole process is incredibly inefficient. I mean, maybe only 1 out of every 10,000 bacteria will actually grab a plasmid. So you're looking at your test tube and you've got millions of bacteria that did nothing, and just this tiny little fraction that actually got transformed. How on earth do you find those few successful ones? And this is where the genius of the design really comes in. This is where those secret passwords, the antibiotic resistance genes, do all the heavy lifting. See, on a normal petri dish with no antibiotics, everything grows. You just get a big smear of bacteria. You can't tell anything apart. But if you add the antibiotic ampicillin to the dish, it kills every single bacterium that didn't pick up the plasmid. Only the successful ones, the ones holding pBR322 with its built-in resistance gene, can survive and grow into little colonies. It's just a beautifully simple and powerful way to find a needle in a haystack. So because of its clever design and just how well it worked, pBR322 didn't just become popular, it totally took over. The plasmid was shared freely between labs, it became the go-to standard for a whole generation of biologists, and it really laid the groundwork for the entire biotech industry. The industry that would eventually give us mass-produced insulin, life-saving vaccines, and so much more. I mean, the impact is just hard to overstate. In science, one way we measure impact is by seeing how many other scientists build on your work. The original paper from 1977 that described pBR322, it's been cited by other researchers more than 3,300 times. That number makes it one of the most cited papers ever published in its journal, which is a pretty clear sign of just how foundational this tool really was. And its legacy wasn't just about its own use, either. pBR322 became the basic model, the blueprint for a whole new generation of better tools. Scientists took its core design and they just kept improving it, creating plasmids like PUC19. This new version was smaller, way more efficient, and had a ridiculously high copy number, making hundreds of copies per cell instead of just 20. It was technological evolution happening right in the lab, all built on the foundation that pBR322 laid down. And here's a fun little piece of trivia for you. The influence of pBR322 actually spread beyond the lab and right into pop culture. In Michael Crichton's book, Jurassic Park, there's a scene where they show a computer screen with a snippet of dinosaur DNA. Well, a couple years later, a scientist at the NIH got curious, ran that sequence through a database, and guess what he found? It wasn't dinosaur DNA at all. It was just a few random fragments of good old pBR322. So pBR322 was the undisputed dream machine of its time. It solved a massive problem and basically kicked the door wide open for decades of discovery. It really makes you wonder, doesn't it? With these incredible new technologies we have now, like CRISPR and synthetic biology, what are the dream machines being built in labs today? And what huge problems are they about to solve for all of us?